Good evening, students. Last lecture we have discussed pharmacokinetic types of drug interaction, where one drug, right, was changing the absorption of another drug, right, and in that various mechanisms we have discussed with clinically important example. Today we are going to discuss the pharmacokinetic interactions which are resultant of alteration of distribution, metabolism and excretion. Okay, so starting with the drug interactions, right, where alteration of distribution is observed, right. So only one category in this case is protein binding drug displacement, right. You must have learned the concept of the protein binding, right, in the general pharmacology, pharmacokinetics, right, definitely you must have gone through. And today we are going to discuss the drug interaction which could be because of this property, right. So you know that once a drug is absorbed, definitely it is now there in the plasma, and you know that you have various proteins, plasma proteins, right, that are present definitely. So that is albumin, alpha acid glycoprotein, right. These are some of the examples, major examples of plasma proteins, where your drug, because of its property, right, chemical property, it could interact, it could reversibly bind with that proteins, right. So there are two possibilities, right. Either that drug is in a free concentration, right, it is not bound to the plasma protein, and second, it is bound to the plasma protein, right. Such type of interactions are very important, where the drugs are having the protein binding more than 90%, plasma protein binding is more than 90%, right. Say for example there are 100 molecules, and in that case definitely 90 molecules of the drugs in plasma, they are bound to the, you can say that, plasma proteins, and only 10% of the drugs that are available in free concentration. You know that once a drug is bound with the protein, it is a large complex. Say for example this is your drug and this is your protein. In this case this is a macromolecular structure, and you know that molecular size, molecular weight of proteins is high, and this high, you can say, weight and you can say size does not allow it to cross the cell membrane barrier. That means it will be remaining inside the blood circulation, right. Drug which is bound to the plasma protein will remain in the plasma unless and until it is available in a free form, right. Or in a short, you can say that drug which is bound to plasma protein is pharmacologically inert. Why I am using this word pharmacologically inert? Because this drug cannot be distributed to another compound, because it cannot cross this cell membrane barrier. Okay. Second thing is, once it is not able to cross this barrier, it will not be able to filtrate also, right. Glomerular filtration is also not possible, so excretion is also not possible. It will not be, you can say, reaching to the liver also, so metabolism is also not possible, right. And definitely it will also need not reach to the target organ, so in that case pharmacological action is not possible. So for a protein bound drug, it is not possible, right, to go in the target organs, in the organs where the metabolism is possible, excretion is possible. In short, it is pharmacologically inert, right. So these peculiar properties are definitely considered while designing the dose of a drug, right, on the basis of those drugs which are having high plasma protein binding, right. Definitely the dose is very much affected by this property, right. So in that case definitely if a drug, right, concurrent administration of a drug which is binding to the same plasma protein, right, which is binding to the same plasma protein, if it is given, and in that case definitely there will be a competition between these two drugs for binding into protein, right. Say for example there is only one chair and there are two person, right, who wish to sit, right. So definitely in that case the one who is definitely is more in strength, that will occupy the seat, right, and other will definitely watch. So same thing will happen if you give drug B, right, then definitely the, because it's a scenario that we are considering competitive displacement, right. Competitive displacement means just both are having binding to the same site in this plasma protein. Okay, we will also discuss this scenario of non-competitive displacement, but here we are discussing a scenario where there is only one site, right. Say for example this is the protein and you have this drug also which was binding to this plasma protein, and you have, say for example, this drug also which is also is having binding, right, to the same plasma protein. So definitely only one drug can bind with this protein, and other drug definitely will be free if it is been set free, right. So remember, definitely there is an increase in plasma concentration, definitely there will be now possibility of more metabolism, more excretion, right, more distribution towards the tissue, right, where wanted, you can say, or desirable organs, right, desirable effects and undesirable effects, right, both definitely will increase. But this free concentration was not expected to be released in the plasma. Now you will say, sir, what effect it will make or what difference it will make that if a small concentration of drug is freely, right, is now available as a free concentration in the plasma. Consider that your drug was given in a dose of 5 milligram, and there is only, you can say that what you can say is a 10% increase, right, or maybe say for example 0.5 milligram concentration is now available more, right. We are definitely discussing in plasma, so you need to consider per ml or per dl, right. So and definitely it will not be this high milligram, I have just written here, it could be in micromole, right. But then consider that there is just an increase of 0.5 milligram, right, considering that the overall dose was 5 milligram. So you will say that 5 milligram will not make a difference, definitely it will not make a difference if your dose of a drug is somewhere around 500 milligram, then 0.5 will not make a difference. But here the dose of a drug is 95. In such a case, 0.5 milligram increase is equal to 10% increase, right. The overall effect is 10%, right. So similar scenario you can consider, and in that case definitely 10% increase in the plasma concentration of a drug will matter, right, will lead to definitely toxicity, right. So this is a very simple example of protein binding drug displacement, right. So free concentration of drug if increase definitely could lead to appearance of an toxicity, right, augmented type of an toxicity that you can observe, okay. And very well example that we have taken, right, that if concentration, right, plasma concentration is definitely is closely to be monitored, right, in some drugs, definitely in that case it will be, right. So remember that this competitive type of drug displacement, right, it depends on two property, one is affinity and second is percentage protein binding, right, and this we will discuss very soon, right, within few minutes we will discuss, right. So that two properties definitely we are going to discuss. Right now we are discussing one more type of an plasma protein displacement, that is non-competitive. Non-competitive definitely you can I am just showing a diagram, right. So here is a diagram, right. So consider a case that your drug was previously acting on this, definitely, right, your one drug is definitely binding here, right. So and another drug, right, precipitant drug you have given, so it is not binding to the same site, it is binding to different protein binding site, but then binding of this drug will change the configuration, right. You can just say that will lead to alteration in the structure of a protein, and this change will now not allow the binding of this drug, and such a case this drug now will be available, you can say that, more in a free form, right. So plasma concentration of this drug will now rise, and such a case it will lead to toxicity of this drug, right. So this is called as a non-competitive displacement. Competitive displacement definitely it occurs at the same site, okay. So with this example, such type of, you can say, interactions are also possible, right, non-competitive displacement where they are difficult to predict, okay. Yes, so two properties as I was saying, that percentage protein binding and second is affinity, right. So percentage protein binding, very simply you can just say that, and most of the time as a student you have this concept, say for example one drug is having 90% plasma protein binding and second drug is having 99% plasma protein binding. So what you will say that definitely this drug which is having 99% protein binding will displace the drug which is having 90% protein percentage protein ability, right. So in that case it is not right. Definitely we have discussed that this type of interactions are only clinically important if the overall percentage protein binding is more than 90%, that is very true. But considering that both drugs are having more than 90% plasma protein binding, then you need to consider affinity. If this drug which is having 90% protein percentage protein binding is having more affinity as compared to this 99% plasma protein binding drug, then in that case it is possible that this drug, right, will bind to the plasma protein and the replaced drug will be this one, right. So it will lead to increase in the free concentration of this drug. Okay, so sometimes we just say, for example, when two person are fighting and one person is definitely, it looks like that the physics of that person is in favor, that yes, he will definitely win, but then also we sometimes observe that yes, it is not always a case, right. So affinity you need to consider in every case, right. So these two are very important. So definitely you must have learned this somewhere in the general pharmacology concept. Okay, let's quickly move to the examples of this type of drug interaction. Okay, and very important examples I have narrated. All of these are very important. I am starting with this one, right, that is phenylbutazone. You know that these are the example of NSAIDs, non-steroidal anti-inflammatory drug, and tolbutamide is an example of first generation sulfonylurea drug, right, which is anti-diabetic drug. Both drugs definitely have a high plasma protein binding, right, and in that case phenylbutazone, if it is given with tolbutamide, this, because it is having more affinity, it will bind and it will lead to, you can say, the free concentration, increase in the free concentration of tolbutamide, right. You know that it is a sulfonylurea category drug, so that means it is insulin secretagogue, right. Insulin secretagogue means it increases the secretion of insulin, and you know that if a higher amount of insulin is secreted in the blood, what it will lead to, it will lead to hypoglycemia, and if plasma concentration of tolbutamide is higher, there is always a chance that this will result in a hypoglycemia, and this hypoglycemia can result in coma also. So if these two drugs are given concurrently, it may increase the action of tolbutamide. Such a case, hypoglycemic coma could be observed, right. So this is the first important example. Same thing you can consider for salicylates and warfarin, right. Salicylates, you know that these are again non-steroidal anti-inflammatory drug, right. Aspirin is the best example that you can consider, and warfarin is definitely anticoagulant, right. So here also the affinity of NSAID is more, so definitely salicylate will displace warfarin from its binding site, and such a case warfarin concentration, right, plasma concentration of warfarin, right, free concentration will increase, and this will lead to increase in the anticoagulant effect, and if it is an a situation where bleeding occurs, then definitely hemorrhagic condition, or maybe say for example increase bleeding, right, that could lead to very, you can say that complications, right, fetal complications that could lead to that could arise because of such type of a drug interaction. Here also you need, if you just closely look, right, then there is also one more possibility, right. Say for example aspirin in a low dose, you know that also is antiplatelet, right. Warfarin as such is an anticoagulant, and salicylate, that is aspirin, is antiplatelet, and that also will delay the blood clotting, right. So pharmacodynamic interaction also you need to consider. Sometimes it is possible case that the drug interaction, right, they are pharmacokinetic also and pharmacodynamic also, right. So here dual mechanism you can consider for salicylates and warfarin, right. So very important example, those patients who are receiving warfarin should not be given aspirin. Okay, similar consideration you can give for valproic acid, phenytoin. Both are the drugs which are belonging to the category of anti-epileptic drugs, right. And here definitely valproic acid, it displaces the phenytoin from its plasma protein binding site, so resultant increase free concentration, right, of, you can say that phenytoin, right, could be observed. Now this is very detrimental because up to a level, right, up to a therapeutic level, phenytoin in its metabolism it increases, it follows the first order reaction, right. So that is definitely with the constant increase in concentration, metabolism also will increase, but after a level the metabolism becomes saturated, right, that means it's a zero order reaction. Okay, and you know that mixed order reaction wherever has been there, definitely it is very, because now metabolism is saturated, further increase in the metabolism with increase in free concentration of drug will not be observed. In such a case phenytoin toxicity, right, in such situation is very common, right, ataxia, nystagmus, this type of an conditions could be observed with this type of a combination if it is been given, right. Second thing is also very important to note here, that phenytoin is classical example of hepatic macrosomal enzyme inducer, and in that case it increases the metabolism of valproic acid. If it increases in metabolism of valproic acid, action of valproic acid is reduced, right? So sometimes it becomes very complicated. Valproic acid leads to phenytoin toxicity, and phenytoin reduces the effectiveness of valproic acid. Okay, so definitely, and pharmacodynamic interaction definitely you can consider, right? You know that valproic acid virtually acts by everything, by increasing the GABA level, by inhibiting sodium channels, by blocking calcium channel, and phenytoin is a very well example, a classical example of a sodium channel blocker. So such type of an interaction, pharmacodynamic interaction, also needs to be considered, right? Similarly, one can consider the effect of salicylates, right, on the methotrexate, right? Methotrexate definitely is a highly bound in the plasma protein, and once you give with the salicylate, it will displace, right? It is capable of displacing it from the binding sites, and this could result in the toxicity of methotrexate. Okay. Salicylates also interferes with the renal excretion, right? Here it is important to note that salicylate, they inhibit the excretion of the methotrexate also, and in such a case, because they are displacing the plasma from the plasma protein binding site, and second thing is they are reducing their excretion, such a case there is a higher chance that a methotrexate, and so that reason these drugs are never given in a combination. Okay, so this is a very important sort of a discussion that we have made, clinically important example, right, we have discussed. Okay, also you need to consider the states, right, where the distribution is altered because of disease state, right? Wherever there is a condition, what you called as a hypoalbuminemia, right? Hypoalbuminemia, that means reduced concentration of albumin in the plasma, right? There are some disease where the albumin concentration is decreased. That case, distribution of all of these drugs which are having high protein binding capacity, right? So that will definitely alter, and in that case definitely resultant toxicity, right? And the adjustment in dose, right, like phenytoin, like salicylates, definitely you need to consider. Okay, I hope you have understood the concept. Moving towards the next one is alteration of metabolism. Now here two situations we will consider. One, that is enzyme induction, and second, that is enzyme inhibition. Okay, so very simple. If a drug induce the enzyme which is responsible for metabolism of the another drug, or in very rare situation it could be a increase in the metabolism of the drug itself, right? So that situation definitely we will consider it as a enzyme induction, right? These are very rare situations. Say for example, some drugs like carbamazepine, there you can observe such type of effect. Carbamazepine, on the consequent doses, right, when you take multiple doses, it increases its own metabolism, and definitely dose adjustment regarding that definitely need to be considered. Okay, so that is one is enzyme induction, right? So consider a case, right, that one drug is inducing this enzyme, hepatic macrosomal enzymes, let us say CYP450, right, hepatic macrosomal enzyme. But remember that enzymes are ultimately protein, and protein synthesis in your body is not instantaneous, right? It will take some time. Yeah, these are some of the examples. Right? All classical anti-epileptic drugs, old anti-epileptic drugs, phenytoin, carbamazepine, barbiturates, benzodiazepine, valproic acid, they are all enzyme inducers, right? Classical hepatic macrosomal enzyme inducer, which we will also see few more examples, like rifampicin is also a very important example, but then definitely the examples we will check later on. One thing definitely you should note that carbamazepine is a rare example where it induces its own metabolism, right? So that definitely we have discussed. Yes, what we are discussing is enzyme induction is not an instantaneous process, right? Because it is involving protein synthesis, so it may take some time. Okay, so let's check. Very simple. Here is a case. Drug once been given, it reaches to the liver, right? Liver is a major organ, right? Metabolism could occur at other organs also, definitely metabolism could occur, but major metabolism of a drug occurs in case of liver, right, where you find two mechanisms, phase 1 and phase 2, right? I am not going in that details, but here consider an enzyme and then definitely this will result in metabolite. Some cases it could happen that metabolite retains the activity, but majority of the case metabolite or metabolites, they have reduced pharmacological action. Okay, right. Some rare example it is also possible, like prodrug, that original drug is inactive and metabolites are active. There the scenario is different. We are considering here a general scenario where drug is an active molecule and metabolites are less active or not active, right? That scenario we are considering. So consider a drug that phenytoin is given, right, and then definitely it is a hepatic macrosomal enzyme inducer. It will increase the number of enzymes. Once these enzymes are available more, metabolisms, drug metabolism will be higher, and the concentration of, you can say that, these metabolites will be higher. Such a case, pharmacological activity, resultant pharmacological activity will be reduced, right. So this will result in, you can say that, loss of therapeutic action, right, when these drugs are given in combination, right, definitely, as compared to if it is given as such, right, combination will result in loss of therapeutic action. Okay, so this is what increase in the hepatic macrosomal enzyme, or induction of the hepatic macrosomal enzyme can lead to. Fine. Similarly, yeah, this is the case that I was discussing, that it is not an instantaneous process, right, because it is involving protein synthesis. So up to a 3 week of, because it could, the activity, right, enzyme induction activity could be start by 3 or 5 days, but maximum effect of enzyme induction could be observed, right, optimally, right, that is 3 weeks, right. Remember that all cytochrome P450 enzymes are inducible, but CYP2D6 is not inducible, right. Definitely CYP2D6 are important, right. Say for example, many substrate like amitriptyline, codeine, right, then propranolol, right, tramadol, these are some of the substrate for the CYP2D6 enzyme. And you can observe many examples, right, which are acting as an inhibitor of CYP2D6, but there are no example of CYP2D6 inducers, right, because the reason is, as I told you, that it is a not inducible type of an enzyme, right. Important category in this enzyme induction is CYP3A4. We will check the statistics of that also in later slides. Enzyme inhibition, definitely just opposite to enzyme induction. Here there is a decrease in rate of metabolism, right, because the, you can say that, the enzymes are inhibited, right. You have given two drug and one drug is inhibiting the enzyme. If enzyme is inhibited, definitely metabolism is also stopped, and if metabolism is stopped, definitely it will result in the toxicity or increase in the action of the object drug. Okay, so in enzyme induction, definitely it will result in loss of therapeutic action. In enzyme inhibition, it will result in the increase in its toxicity. Fine. So here also examples have been there, right. So you can just see that verapamil, disulfiram. Disulfiram is an example where it is not inhibiting the hepatic macrosomal enzyme, but then we are considering it as an example. You can say it is definitely hepatic enzyme, but it is not CYP450 enzyme, right. So disulfiram, you know that it is used as an antabuse, and definitely it inhibits aldehyde dehydrogenase enzyme, right. That is how it is used, because once the alcohol is converted to aldehyde, and then finally from aldehyde it is normally it gets converted into acids, but if you give disulfiram, then definitely the metabolism of acetaldehyde into acetic acid will not happen, and such a case definitely accumulation of acetaldehyde will result in vomiting, so many symptoms you can consider, right, and gradually patient avoids the use of alcohol, right. But this disulfiram definitely inhibits the metabolism of many important drug also, right. So that we need to consider. Inhibition of enzyme is definitely an instantaneous process, right, because just you can say reaction between the substrate inhibitor, right, with this enzyme. So definitely within a short, you can say that period, that is within 24 hours, the such type of drug interaction can be observed. Okay, yeah, this was this is the what effect that I was discussing. Most of the drugs, you can just see that they are you can say metabolized by CYP3A4, right. So most of the clinical drug interaction which are because of the change in the metabolism pattern, they are belonging to, most of this they are belonging to this category, right. So substrate, definitely here you can consider carbamazepine, diltiazem, some of the important drugs like HIV protease inhibitors, right, ritonavir, right. Similarly, you can say that HMG co-reductase like lovastatin, simvastatin, right. These all drugs definitely are substrate. And inducer of CYP3A4, they include grapefruit juice. Then this grapefruit juice is definitely inhibitor. Inducer will include carbamazepine, phenytoin, rifampicin, even herb like Saint John wort, right, barbiturates, right. These are some of the examples of CYP3A4 inducers, right. Inhibitors of CYP3A4 include clarithromycin, erythromycin like drugs, right. So those are macrolide antibiotics. Then you have fluconazole. Again, you can consider example of food like grapefruit, right. Then you can have ketoconazole, right. So these are some of the examples of inhibitor of CYP3A4, right. You can find this list of drugs in you can say that Remington, right. Remington volume 2, right. You can definitely go through, and these all examples are given, right. Rest of the enzymes are also important, right. I know that it is difficult to remember these examples individually, but then if someone is preparing for GPAT examination, definitely such examples they need to remember. Okay, so this is all about rifampicin. Is again an example that you can consider is an hepatic macrosomal enzyme inducer, and it induces many enzymes, right. So rifampicin induces many enzymes like CYP2C19, right, then CYP2C9, right, as well as CYP3A4, right. So rifampicin many a time is been you can say shown, right, clinically been shown to have drug interaction, and that reason is because it is increasing, or it is inducing a wide range of CYP450 enzyme, right. So these are three examples that I have shown here. Okay, so moving toward the next one, right, that is your examples, right, of this drug, right. Most of the case we have discussed, important one we have discussed, but still this is a list that you can refer to, right. So these are the example of enzyme, hepatic macrosomal enzyme inducer. So barbiturates, phenytoin, St. John's wort, rifampicin. Cigarette smoking also you can consider here. Cigarette smoke induces CYP1A2, right. So CYP1, I am not writing CYP, right. So CYP1A2. Someone was asking last lecture, right, that how smoking increases the metabolism, right. So that is definitely it is by enzyme inducing property, and it specifically increases the proportion of CYP1A2, right. So by that virtue definitely the metabolism, and this is very important, that drug like theophylline, right. Theophylline is a substrate for CYP1A2, right. Theophylline, you know, that is a bronchodilator and is given in asthma, right. And if the patient of asthma is also concurrently having a cigarette smoking, definitely two detrimental effect will be. One thing is definitely smoking is dangerous, right. It is definitely not advisable, right. It is advisable for the patient of asthma to quit smoking, right. And second thing is also cigarette smoking will increase the metabolism of theophylline, and that case resultant plasma concentration will be less and bronchodilation also will be less. Okay, these are examples of inhibitors of enzymes, and you can just see the example, right. So these are some of the examples, right. Protease inhibitors, MAO inhibitors, fluconazole, isoniazid, these are very important examples which you need to remember. As I told you again also, for individual examples, definitely for individual enzymes, hepatic macrosomal enzymes, the variants which you have checked in the last slide, you definitely need to go for an example. If you are not able to remember all, then please at least remember the examples of CYP3A4, right. Why I am insisting is because the wide range of drug will be covered, that is one thing, right, where clinical interactions are found. And second thing is there is also one more topic which we are going to discuss today, that is P glycoprotein, and that case also P glycoprotein also definitely there is an overlapping substrate specificity, right. That means the drug which is inducer of CYP3A4 is also inducer of P glycoprotein. A drug which is inhibitor of CYP3A4 is also most of the case inhibitor of P glycoprotein. Fine. So that is what you need to remember. Moving toward the next is alteration of excretion, right. Until now we have completed alteration of the distribution and alteration of metabolism. Now we are moving toward the alteration of excretion. So there are various mechanisms which could lead to alter in excretion of one drug, right, by concurrent administration of another drug. Let's see an example. First one, that is by alterating the pH of the urine. If you remember, then in absorption we have checked that if a drug is ionized, definitely it is not able to cross the cell membrane barrier. If it is non-ionized, it is able to cross the cell membrane barrier and it is able to diffuse easily, right? That is what I am trying to say. Same concept you can consider here. So if you alter the urinary pH, definitely you are also making the proportion of ionized and non-ionized drug, right, present in the urine also is going to be changed, right? So one thing that is going to be changed is the more the non-ionized part, more is the water solubility, and more is the water solubility, higher is the chances of excretion, right? You know that drug, if it is more water soluble, excretion will be higher, right? Second thing is, if a drug is non-ionized, say for example drug is present now in the this lumen, right, lumen of proximal convoluted tubules or distal convoluted tubule, and if it is in, you can say, the ionized form, it cannot come back through the diffusion. If it is an definitely non-ionized form, lipid soluble form, it is a possibility that it could come through the diffusion, right? So both case definitely excretion will increase, right? So rate of excretion, it is dependent on the water solubility, and ionized drug is more water soluble. Second thing is, once the drug is in there in the lumen, right, where the your glomerular filtrate is there, then diffusion is also difficult because drug is in the ionized form, right? So salicylates you can consider, right? Say for example, if urinary acidifying agents are given, right, concurrent administration of drugs which can decrease the pH of urine, then salicylates, you know that these are weak acids, and here the pH of urine is also definitely now acid because acidifying drugs. So that case, salicylates now will be non-ionized form and the urinary excretion of salicylate will be less. Such a case, resultant toxicity of, you can say, or increase in the plasma concentration of salicylates could be observed. Or another end, this concept can also be used to treat toxicity of aspirin, right, salicylate, that is aspirin. Say for example, there is an acute toxicity of aspirin. One of the approach, right, to reduce the toxic effect is make the pH of urine alkalize. Okay, if you make the urine pH alkaline, definitely now salicylate, because these are weak acids, in presence of basic pH, it will result in more ionized form of the salicylate. More is the ionized proportion, more is the excretion, and toxicity of salicylates can be reduced, right? So this is an approach that is used in management of toxicity of aspirin. Okay, urinary alkalizing agent, you can remember definitely sodium bicarbonate is an example. So NaHCO3 definitely you can consider. And second example that you can consider here is you can say that drugs which are carbonic anhydrase inhibitor, right? So carbonic anhydrase inhibitors include acetazolamide, right? So such examples also you need to remember, right? So a clinical important drug interaction that you can consider in such important situation, right? Many of the diuretics, they also have important effect on the pH of the urine, and all of that definitely you can discuss here, right, under the heading of alteration of urinary pH. Similar situation you can consider drug like dextroamphetamine, and alkalizing agent, and if definitely this is a weak based drug, right, amphetamine, you know that, right, this is actually nowadays it is banned, right, so it is a drug abuse, and in that case if alkalizing agents are given, amphetamine is retained in the body for longer duration, and such a case toxicity of amphetamine will be well evident in those cases, right. Moving towards the second mechanism in the alteration of excretion is alteration of active transport, and a very well famous example, penicillin probenecid, right. You can consider this as a beneficial type of a drug interaction. You know that both of these drugs, they compete with the same site for the, you can say that active secretion, right, and definitely you know that there are three processes in urine formation, right, that is, one is glomerular filtration, second is tubular reabsorption, and third is the tubular secretion, right. So the excretion of penicillin is largely, it is exclusively through the tubular secretion, right, and in that case probenecid also is excreted by this you can say mechanism only, right. Probenecid is a uricosuric agent which is used in treatment of gout. Now if you give these two drugs in combination, both will compete for the same site, and probenecid definitely will be secreted, right, because it is having more affinity for those transporters, right. Remember, tubular secretion is always an active mechanism, it is never passive. Such a case, probenecid will be excreted first and there will be retention of the penicillin. This will increase the biological half-life of some of the important penicillins where the biological half-life is an issue. Okay, and such a situation definitely for treatment of disease like syphilis, gonorrhea, right, which are sexually transmitted disease, you need prolonged you can say exposure of a drug, right. So and continuous exposure of a drug, right, surrounding the bacterium, right. So then and then effective eradication is possible. So definitely penicillins are combined with probenecid, right, because of this such an important pharmacokinetic property, right. I hope it is clear. Similar scenario, right, definitely you can consider, right, for NSAIDs and methotrexate, which we have discussed earlier under the plasma protein binding, right. So in that case we have also said that NSAID reduces the excretion of methotrexate, and definitely it is through the alteration of active transport. Okay, moving toward the you can say that next you can say category, right, that is alteration of drug transport, right. Until now we have completed ADME, right, alteration of absorption, distribution, metabolism and excretion, right. So this is today's last discussion, right, of a topic of that is we are going to discuss, right. So you know that there are some very important transporter molecule, right, and one of that important transporter is P glycoprotein. P glycoprotein is important in you can say many you can say by many mechanisms, right. Say for example, these are some of the drugs, right, which are actually affected by the presence of P glycoprotein, right, and in that case P glycoprotein is having two important role, right, that you can consider, right. Overlapping substrate specificity is there, that we will discuss later on, but the function of P glycoprotein, that is what we are going to discuss, right. So first is it is definitely a transport system, right, and this transport system definitely it acts as a barrier for many important pharmaceutical agents, like say for example loperamide does not have a central nervous system. You know that loperamide is an anti-diarrheal drug, right, it is commonly prescribed in India for diarrhea, right. In many countries it is banned, but definitely in India it is used, and it belongs to opioid type of an category, right. So it's an opioid drug, and normally you know that opioid drug they have a very prominent CNS effect, but loperamide does not have a CNS effect, right, just like morphine, right, it does not have any effect, euphoria and all that. So in that case, why it is not having morphine like effect? The reason is P glycoprotein does not allow its transportation across the blood brain barrier, and that is how the effect is only in the GI tract. You know that opioid drugs they have major effects in two organs, right, one is CNS and second is GI tract, and if CNS effects are definitely absent, then loperamide is a good choice, right, to reduce the GI motility, right, definitely to reduce the you can say diarrhea, right. But definitely as I told you, it is banned in many countries because of the cardiovascular effect. Okay, forget that. Chalo, focus on the P glycoprotein. First thing is it is a transport system that act as a barrier. I give an example, loperamide. Second thing is it is acting as a pump that sometimes faces. These are two contradictory discussion that we are making. At one point it is acting as a barrier, right, that is also a possible case, but in another case it is facilitating the transportation of some of the drug. Okay, and one of the example of this is digoxin, right. So digoxin transportation is facilitated by this presence of P glycoprotein. Remember that we are discussing here drug like diltiazem, atorvastatin, digoxin, where the dose is quite low, right. See, if the dose is comparatively higher, right, if it is in few hundred milligrams, right, in that case definitely alteration of drug transport will not make a matter, but those type of a drug where the dose is definitely lower, there you can consider clinically important example, right. So that definitely you need to consider. So remember that digoxin, diltiazem, atorvastatin, right, these are all you can say that drugs where presence of P glycoprotein is very important, right. And consider a case that if P glycoprotein is inhibited, then, right, say for example I am giving a drug like quinidine. Quinidine is P glycoprotein inhibitor. So in that case definitely now this digoxin will not be facilitated, right, the transportation will reduce. Such a case definitely effect of digoxin can be reduced, right. So this is the role of, right, once you are knowing the role of P glycoprotein, then examples and their interpretation are definitely very important, right. I already told you that digoxin is transported by P glycoprotein, And these are some of the examples of the P-glycoprotein inhibitors, right? Quinidine as well as verapamil, both inhibits P-glycoprotein. Okay, if they inhibits P-glycoprotein, the serum drug concentration, right, of the digoxin gets reduced. If it gets reduced, definitely you know that's a narrow therapeutic index drug. Loss of therapeutic effect could be observed, right? Similarly, you can take an example of loperamide. Loperamide does not produce morphine-like effect because P-glycoprotein is acting as a barrier, and in that case, loperamide is not able to enter the CNS. But if you give a P-glycoprotein inhibitor like erythromycin, ketoconazole, quinidine, this loperamide will also let you feel that you are king of the world, right? Morphine like effect, euphoria like effect, that will be observed with loperamide, right? So I hope you got the idea, right? So this is all regarding the alteration of drug transport. Very short description is now remaining, but this definitely we will discuss in the next lecture, right? Well, we will discuss the important pharmacodynamic interaction. Also, we will check that you can say that how to reduce type of a drug interaction, and we will also have a brief overview of different methods of drug interaction, right? How to detect the drug interaction, that also we are going to discuss. If you have any doubt, definitely you can ask me. Thank you very much.